Okay, so we will go fast. And we will talk about an update about Reid on robust ancestry inference from challenging human molecular data. So it's well known from epidemiology that there are links between diseases and ancestry. So example, we have prostate cancer is more, the incidence is higher in African population, leukemia more frequent. The incidence is higher in European population, and not just in cancer, but example for Alzheimer. Alzheimer have a higher incidence in African ancestry or Hispanic population. So the ancestry probably linked to the disease via environmental component and a genetic component. We are interested in the genetic component. So it's important to have a good genetic ancestry, not just the self-declared ancestry, because yes, to get the real genetic ancestry. So it's well known how to infer the genetic ancestry from genotype, from cancer-free whole genome, exome, microarray, can do clustering following dimension reduction like PCA, likelihood maximization like admixture, and multiple other methods. But there is a double challenge. We can we infer the ancestry using only cancer-derived genome, or from a protocol other than the whole genome or whole exome, like RNA-seq, ATAC-seq, small panel, ChIP-seq, bisulfite sequencing. And if we have access to this type of data, we unlock a vast data resource from the public database, example, GEO and SRA, to study the ancestry link to the disease. So we published our method in 2023 in Cancer Research, method Reid, and in Bioconductor in 2023. Currently, Reid run on global ancestry for whole genome, whole exome, cancer genome, and gene panel, RNA-seq, single-cell RNA-seq, ATAC-seq for cancer too. In progress, we are packaging a version with a mixture. We're working on local ancestry, low coverage, whole genome, ChIP-seq, Xintong here, work on ChIP-seq and cytosine converted or bisulfite sequencing for whole genome or IRBS. And we are soon have a Galaxy version of Reid. So how it works, we start from a population reference. By default, it's 1,000 genome. We have the different continental ancestry for the global ancestry, African, European, East Asian, South Asian American. And so Reids take two input. The first input is the molecular profile from RNA-seq, WES or whole genome or gene panel, etc. And the population reference. We generate what we call data synthesis, and we'll talk a little bit better about it after. And we optimize if need, if the inference method need optimization parameter, and assess the performance if the method is able to infer accurately the ancestry from this specific molecular profile. And finally, we infer the ancestry. So the data synthesis, the idea behind is we use the specific molecular profile. We extract an individual from the reference. We remove the individual from the reference, and we transfer the genotype from the reference individual in the profile, the profile we are studying, and we keep the same characteristic than the profile. So we are able to know if we are able to call correctly the ancestry on this profile, specific profile. And we run this multiple times. So the performance based on synthetic data, here Reid by default use PCA with key nearest neighbor. So here we have two parameters, the number of dimensions used and the key. And here is the accuracy obtained for different methods. So it's near 1 for whole genome, whole exome, concert, and RNA-seq, and very good for gene panel and single-cell RNA-seq and antac-seq. In development with Xintong, we have DNase, ChIP-seq, and bisulfite sequencing, RBS or whole genome, and they are really good too. Some exception for some target of ChIP-seq didn't work well. If we look at the cohort performance, so if we compare the ancestry call based on cancer sample or panel sample, RNA-seq sample versus the cancer-free sample, we get a very good accuracy for TCGA breast cancer. For a subset of TCGA breast cancer, we are near 1. For leukemia, beta-AML, we are again at 99% accuracy. And for TCGA ATAC-seq from multiple cancer, we have 62 samples. We are near 1 again. For the ChIP-seq, is very good too. We use the Rush Alzheimer's disease from ENCODE, and we compare, in this case, the ChIP-seq with the ancestry call on the RNA-seq. We have near 1 for near all targets except the H3K27M3, which is 92%. The bisulfite sequencing, RBS, are very good too. We use a Ewing sarcoma. I put the SRA project ID and Crohn's disease to validate it. So we have after we implement, we switch the PCA for admixture and study the admixture of the sample. Here we use as an example TCGA breast cancer. Each column is a case, a profile. We have the color represent the proportion of the ancestry. So here we have the cancer-free whole exome and the tumor and the RNA-seq. For the same column is the same sample. We saw that they have essentially the same profile, but they have a little bit more noise for the two tumor case. And if we want to use a metric to do it, we use Jensen to compare. We use the Jensen-Shannon divergence, compare the two admixture distribution, and we saw for the synthetic data, the Jensen, the GSD is smaller than the random one. And if we compare cohort-wide for the whole exome, cancer-free versus tumor, it's again very small for the and in conclusion, we develop an accurate data tool, and the data set is unable to let us evaluate the accuracy of the ancestry. And we have the implementation in Galaxy soon and the packaging with the admission. Thanks to everybody.
Okay, so hi everyone, my name is Katherine. I work with the Galaxy P team at the University of Minnesota, and I will be presenting about Meta-iPep, a meta-immunopeptidomics pipeline implemented within the Galaxy platform. And we developed this to characterize HLA-bound microbial peptides. So quick at the top of this presentation, I want to quickly acknowledge and thank the Galaxy project and the EU server, since that's what we're using to develop and test this pipeline. For funding, we are supported by NIH grants, as well as a grant to the Masonic Cancer Center at the University of Minnesota. And thank you to my group Galaxy P at the University of Minnesota, especially the late JJ for all of his amazing contributions, as well as the Nesvizhskii lab at the University of Michigan for their work on FragPipe, which we use in this pipeline. So up top, I'm also going to give a brief background on antigen presentation and lead into how we use that for metaimmunopeptidomics. This will be a broad overview. I will also be presenting a poster on this, but to respect the time, this will be very general. So our immune system is alerted to threats through our T cells recognizing foreign antigens. Antigens are essentially molecules that are deemed as non-self, and these trigger your immune response. These peptides can be grouped into two types, those that bind to HLA class 1, and the other group are those that bind to HLA class 2. And the difference in these lies in where these antigens come from. For HLA class 1, these are endogenous, so these are coming from within the cell, or exogenous, which means they're coming from outside the cell. Looking at microbial antigens in particular, these would mean intercellular pathogens, meaning like a virus that hijacks a cell's machinery. And so we would take antigens in this context would be viral antigens or bacterial antigens, like Mycobacterium tuberculosis that causes tuberculosis. And then class 2 is extracellular pathogens. So something like a staph infection, you would get peptides from Staphylococcus aureus. And so how does our immune system actually get alerted? We use T cells. For HLA class 1, CD8 plus T cells are also called killer T cells, recognize class 1. They're called killer T cells because they directly destroy infected cells themselves. Class 2, HLA class 2 molecules are recognized by CD4 T cells, and they're called helper T cells. That's because they don't destroy cells themselves. They actually send signals for other immune cells to come help out with that. So moving into how this ties into Galaxy and metaimmunopeptidomics. Also, it is a mouthful, but I will try my best to break it down. So metaimmunopeptidomics, the immunopeptidomics is, as I mentioned, we're studying peptides that trigger the immune system. The meta part comes from metaproteomics, which is the study of microbial proteins and peptides. Mass spectrometry is the manner in which we analyze samples. And so to put it all together, we are using mass spectrometry to look at because we can get a better understanding of microbes and their role in human disease, and potential targets for improved treatments that could account for different microbial contributions. So I'm going to quickly go over the sample prep and then move into the bioinformatics phase. So first, cells are lysed. Then we immunoprecipitate peptide and MHC complexes. We only want the MHC-bound peptides. We wash away the unbound peptides. These peptides are then analyzed by mass spectrometry. And then here is where Galaxy and the Meta-iPep pipeline is really coming into play. Another issue with immunopeptidomics is that typical mass spec experiments, proteins are digested by an enzyme called trypsin, and trypsin has sequence specificity, and which essentially means that you can sort of limit your search base, because if we know that trypsin only targets certain sequences, then we can sort of constrain our search base. However, the tricky thing about immunopeptidomics is that they don't have that specificity, and so our search space is way larger, which leads to lower sensitivity. And so what we're trying to do with Meta-iPep is to sort of address that issue through our pipeline. And so how Meta-iPep goes is that first we try to predict microbial taxonomy. So this is what we expect to see. And then through tools within the Galaxy program or platform, we identify, verify, and predict HLA binding. So the first module is peptide identification. So first, raw sample data. So this is after the samples have been analyzed by LCMS. We get it from various sorts of human samples to look at human disease. We also have our search database containing microbial, human, and CRAP proteins. CRAP, I don't have it on this slide, but it stands for like basically contaminants. So generally like sort of like any sort of contamination during the sample processing, like say like plastic off of like sample tubing, things like that. So we want to account for those and not accidentally mismatch something. These input files go into a program called FragPipe. And what FragPipe will do is that it'll look for matches between the sample data and the database. And now we will get a big file of peptides, and then we'll filter for microbial. To be brief, we'll focus on one sample, 86B2. So FragPipe first gave 11,000 peptides, and we filtered out to get 92 microbial peptides. Next is the verification step. So the reason why I want a verification step is that because we have such a large database search space, it would benefit from having another tool that would basically double check that did we actually get microbial peptides. So we had to take our microbial peptides and our another database and the sample data, put it into pep query. If I find a microbial peptide in the sample and in the database, well, the database only has human and contaminants, so I'm tossing that not as confident. But if it's not in the database, but it is in the sample, then I have more confidence that it's actually microbial. Sort of skipping forward. So this sort of visualizes like the sort of proportion. So we identified 92 total microbial peptides, but only a small portion actually get verified. And this is sort of the issue of quality over quantity. Like we want to have more confidence, even if it's a smaller proportion of what gets reported. So now we want to get more information. I'm trying to be brief about this. So now we want a taxonomic and functional annotation. And for this, we use two tools called UniPep and BLASTP. To give an example, this is a taxonomy tree. You can see from less specific taxonomy and more specific to species. Now we look at HLA binding. So this is actually looking at which peptides actually bind to what HLA molecules. And so we use two tools called NetMHCPAD and NetMHCN2. Fast forwarding. So this is sort of a visualization of what binding looks like. So darker colors mean stronger binding, lighter colors is weaker binding. This is class 1, this is class 2. There's no biological significance because this is still preliminary data. What's next is we could expand to other samples. Again, metaimmunopeptidomics, where we created Meta-iPep for this. And my poster later today is number 23. Thank you for listening.
Yeah, hey. So I'm a professor at Hopkins. At this conference, I'm part of the Bioconductor team. I'm part of the technical advisory board for Bioconductor. But today, I'm really going to talk about sort of methods and biology. So we have known for a long time that with modern Oxford Nanopore data, we can get long-read data. We can get DNA methylation out really easily, and we can also phase really easily because we have these really long reads. And that was shown by, in some seminal work here from VHI. They did this in F1 mice between two inbred strains, and that I'm basically going to talk about a much larger experiment along this line here. We did it with human data some years ago. We were kind of cheating because it's really easy to phase if you know the parental haplotypes, and we knew that in this particular case. So F1 mice, for the people who are not totally up to date on the genetics, we take inbred strains and then we cross them. We make one generation. And in the F1 cross, there's no recombination that's happened. So all the F1 mice are genetically 100% reproducible, and they're also sort of heterozygous everywhere. So this is like phasing. This is super easy to phase. We can also create F2 mice, and then recombination kicks in. Each mice is unique, and phasing is now much more hard. I mean, locally we can phase, right, but sort of across the entire genome, it's going to be a little bit hard because of recombination. Now, after phasing, which is sort of relatively easy, you then are faced with this problem that, you're actually not really done, because you have to make some comparison across 2 different genomes. Now, these are 2 different genomes, right? And we started this in 2019 for inbred strains, but it works the same for F1 mice. And our method, our approach is sort of based on tracking CpGs that are the nucleotides that carry information about DNA methylation. And we basically have to kind of do a multiple alignment between the 2 genomes. And each CpG, we kind of have to track across the 2 different genomes. We analyze them in something we call an intermediate genome. We have to track each CpG across the 2 genomes. It's a little bit like liftover, except that we have to take, we only care about CpGs. And for each CpG, we have to map it. We have to know whether it maps to a CpG, it maps and it has mutated away from a CpG, or whether it doesn't map. If it's, for example, part of a large insertion or deletion between the 2 genomes, it doesn't map, it doesn't make sense to analyze it. We need to know this, and we need to know the spatial position, because that's sort of part of our method. So that's a ton of bookkeeping, and it's a little bit, there's not a lot of good tools for it out there. Here's a little bit of an example from this old paper, where all I really want to highlight is that one thing we do pretty routine in this space is that we switch the reference genome, we repeat the analysis and switch the reference genome, and then we see if things move around. And if they move around, we don't really trust it that much. So here's a putative DMR that basically flips sign completely, depending on whether or not you align it to the Black 6 genome, align it to the Cas genome. And if you use sort of our method, we kind of deal with it. So we got a little, we used, over the years, we've used sort of different things. We've used a ModMap. That was a tool developed for the mouse community. We didn't like that, partly because it was very hard to input our own genomes in it. Then for a while, we've been using a big mouse cross graphical genomes that is also kind of painful, partly because it's kind of hard to extend it with our own strains. So in the end, after avoiding this for 6 years, we sat down and wrote our own pipeline that's sort of putting together a lot of bookkeeping. We call it Plasma. The input is sort of reads and 2 parental genomes, and then it does sort of this multiple alignment, the phasing, etc., and ends up with like placing everything in sort of a common intermediate genome and analyze it. So conceptually, it's a little bit like WASP for the people who know what WASP is. It's all written by a postdoc of mine, former graduate student at Hopkins. Adam, you can see, is a guy who wants to go somewhere. He asked a picture, and he sent me a picture of the blazer. So we're not actually better at phasing. This graph here basically shows a couple of samples where we compare to sort of 1 genome and use what's happening. We don't really do better at phasing. But what we do better at is that we rescue a lot of CpGs. This is where it's 1 genome, where the 1 genome is a Black 6. We rescue roughly 10% of CpGs in the genome because we kind of account for, we use basically 2 reference genomes for mapping. Okay. So let's look a little bit at some data. That was sort of a little bit of background. We've done this sort of massive mouse experiment in both liver and muscle. In a liver, we have like a bunch of inbred. We have crossed them in all different ways. We have biological replicates. We have 2 mice that we have analyzed with adaptive sampling. And we have pretty nice coverage, right? 20 eggs in each sample. On the phase genome, we have like 13 eggs. And then for this data that comes now here, we haven't used Plasma, but we kind of get the same result. We are using this collaborative cross-graph genome that, on one hand, got us to where we are. On the other hand, we kind of dislike it because it's been really hard to work with. It's been very hard to extend. And some strains that we're interested in are not part of it. So we can analyze roughly 2/3 of the genome. That's because we are using these collaborative cross mice. I won't get into the genetics, but that's sort of expected. That's basically parts of the genome where the strains are IBD. So we can't really phase them. We find sort of around the order of 8,000 sort of standard allele-specific DMRs and around 800 allele-specific DMRs that don't follow Mendelian patterns. That's not really super surprising, but let me talk a little bit about a sort of a standard MQTL. So over here, we have around 40 samples up here. We have sort of a smoothed lines here, and you can really not distinguish them all because there's very little biological variation. The thick lines are the group averages, and there's 4 thick lines. There's the inbred A and the inbred B, and then we have the A allele in the F1 mice. And what you can see here is that it tracks, I mean, you can't really, I mean, they're on top of each other. They track perfectly with each other, right? So this sort of spatial pattern we have of DNA methylation is totally preserved across replicates, and it's also totally preserved when we go into the F1 mice. And if you go into the F2 mice, we also see exactly the same thing here, which is what we would expect for something established by cis regulatory factors. Over here in the F2 mice, because we have recombination, trans factors get recombined in an individual specific fashion. So yeah, this is what we would expect for sort of a standard cis-controlled effect. It's pretty nice to see. We knew about this, of course, but it's really nice to see. I mean, it's really amazing to see how well it looks when we look at it spatially. Here's something that's a little bit more interesting, well, a lot more interesting. So here we have something that's established in trans. So we have the two inbred mice here, the two inbred lines here. And what happens in trans, where there's a copy of each of the F1 mice have one copy of the trans allele from A and one copy of the trans allele for B. So they achieve this intermediate pattern. But then what happens in the F2 mice, where the transacting factors get recombined, so each individual have either 0, 1, or 2 copies of the trans factors, it kind of goes all over the place. So this is an example of a non-dominant transacting QTLs, which can either be something known in genetics as incomplete dominance or co-dominant. That's a little hard to explain, so we kind of need a picture. So here we are breeding two plants. They are kind of white and red, and we are seeing the offspring. So this here is known as incomplete dominance. You can see that the flower petals here have the same color. It's sort of an intermediate color, but it's the same color everywhere. Over here, we have a co-dominant transacting factor, where the flower petals are either white or red in a local manner. Right, so in a local manner, so how does it, how do we even define this thing here with methylation? So methylation happens at sort of the genome level. So we got to look at the individual reads. Here's an example of a co-dominant transacting QTL. So the parents are either all blue, which is unmethylated, all black, which is methylated, and the offsprings here are sort of intermediate, but you can see either each read is either completely blue or completely black, and then some arrows. Over here we have a different allele. Oh, shit. Okay. Yeah. Over here we have a different allele where it's not quite as nice in the parents, but it's like predominantly black, predominantly blue. And then down here in the offsprings, we have that each CBG is almost kind of a mixture in it. I won't say random because I'm not going to claim that it's random, but in a mixture of sort of black and blue sort of mixing together, a little bit like the red and the white thing. So this was work done with Andy Feinberg's lab at Hopkins and David Threadgill for the mice work. And the graphical genome we got from the Macmillan lab at UNC. Yeah, and then if people are really interested in this, I'm just going to put a little plug in for some work I did with a PhD student in Denmark, where we are basically, this is sort of for handling the situation where we want to genotype CBGs, for example, in human. We don't want to run a whole genotyping algorithm. We just want to take the output of ModKit and run on either Nanopore or PacBio data. Then we can genotype the CBGs, get the right CBG state, and analyze it. It's actually pretty nice if you're in this space here, you know what I'm talking about. Otherwise, forget about it. Okay.
Thank you for the introduction. Hi, everyone. In my laboratory, we are studying ovarian cancer, and while studying ovarian cancer, we were interested in studying more the activity of cancer cells. So we were interested in once you identify the identity of cancer cells, trying to understand a little bit more what cells are actually doing. So we can analyze the activity using gene expression signatures. These are biomarkers, so are more like a list of genes whose expression scores can be combined to obtain a final score, and that can be categorical or classifier. And there are many different expression signatures already existing in the public literature. So we just wanted to compute some of them in our datasets. And we figure out that actually it's not so easy to do that, because many of these papers have some hidden steps, hidden files. Sometimes it was really struggling to compute them on your own data. So we decided to overcome this problem. And we wanted to implement a package that is more like a collection of signatures coming from literature. So we collected them, implement in another package to make them be more usable for everybody. So we developed SignFinder. For now, there are more than 70 signatures collected inside it, but obviously we have to maintain it, and it's getting bigger and bigger. And yeah, we started from ovarian signatures, so there are more on the ovarian, but actually then we moved to other type of cancers, so other type of tissues, and also there are 19 signatures that are pan tissue, and they cover different type of activities and topics. Mainly, they are related to the immune system, because you can find more signatures on the immune system in literature, but then they cover other type of topics of cancer activities. It takes in input the summarized experiment data, single cell experiment, special experiment, and this type of signatures, all of them, they return a single sample score that in this case would be single cell score or a single spot score for spatial transcriptomics data. And the first thing that you can do once you compute the signature you're most interested in is to check the quality of that signature for your dataset. That means maybe you want to check the percentage of genes that you actually have inside your dataset. From these genes, how many have zero values? Also, the final scores, how they correlate with the total expression of genes and with the total amount of zeros. And taking together all these three arguments, we created a final goodness score. So you can understand how much that signature is reliable for your dataset. And then there are several types of visualization functions that you can use to better explore the signatures. Some are thought to be single signature, so just plot the trend, if it is related to survival. And also with this plot, you can check which gene are mostly contributing to the final score. Or also you can compare multiple signatures between each other, like for example, see if they correlate. And here I report just a case study. This is a human breast cancer carcinoma that is available from the 10x genomics datasets. And we asked an anatomopathologist to annotate the tissue for us. And he told us that you can see in red here the cancer area, which have some necrotic tissue inside. And it is surrounded in blue, you can see, by cancer-associated fibroblast, but also by high-density infiltrated lymphocytes. And we reported the annotation to the spots, and everything that was around outside the annotated area was just called stroma generally. So I computed all the signatures available in SignFinder for breast cancer and also the pan tissues one. And for example, here I show you these hypoxia buffer signatures. You can see that hypoxia scores are much higher in the tumor area, and with also some area around the tumor with low, medium levels of hypoxia, and then really low scores outside it. Also, I show you this signature. This is supposed to show you the epithelial to mesenchymal transition activity. Here you can see an opposite trend. So you have a really mesenchymal-like status outside the cancer and low scores inside the cancer mass. But actually, if you focus more on the tumor spots that are the ones that I'm showing you here, you can see that actually the the spots on the border tend to have a higher level of mesenchymal signatures, and the spots on the inner part tend to have the lower levels for this signature. You can also see this different trend in this plot where I'm showing you the tumor spots divided by who is their neighboring cells. So, for example, here you can see tumor spots that have 6 tumor spots around them. So in the inner part of the tumor, when tumor cells are surrounded only by other tumors, you can see the lower scores. On the opposite, when the tumor cells are surrounded more by cancer-associated fibroblasts, they tend to have the higher scores. So that's it. We created CineFinder, that is a collection of gene expression tumor signatures coming from public literature. And it has been included in Bioconductor already since November 2022. And you can also find the publication on genomics and bioinformatics. Thank you for your attention, and all to my lab members for the support.
Thanks for a great talk. Questions? We have a minute or two. Yes.
So great talk. So I was kind of curious. So you showed like a trained pathologist annotating like the different cell types, and then you showed the like equivalents that you could find. So I was just curious, how does like how much biological heterogeneity is this? So if it's within the same patient, like are you able to match well with the pathologist annotations in different patients and different diseases? Do you have to like retrain the this classification scheme or?
Yeah, actually, for these samples, we had just one patient. We didn't have different patients. So I don't have the comparison with other literature. But yeah, we actually saw different heterogeneity. You can really explore that here. And you can see that this was just an example of a signature having different type of activities depending on where the tumor cells were located, how much they were interacting with the tumor microenvironment or not. So yeah, we already saw much heterogeneity in a single sample. Thank you for the question.
Thank you. Okay.
Yeah. Okay, thank you for the introduction. I'm Parikshit. I'm from the Jason Shoemaker lab, and we study the host response to viral infection and sex-specific responses. So, so this project I'm going to talk about is concerning influenza A virus, and so influenza infects 8% of the U.S. population every season, and over 500,000 people die around the world per year. As is the case in a lot of like lung immune infections, the key here is to try and have like a balanced host response. So you want to have a sufficient response to kill off infected cells, but not too far where you have a lot of immune cell infiltration, creating tissue damage. And we know that there's ample evidence that like females of reproductive age are disproportionately infected by sort of this higher response. And so what is not known is like sort of this hormonal impact on viral infection. And so the goal of our lab is to use network tools to identify these sex-specific virus replication host factors. And so for this study, we have our primary data set, and then we have a validation data set that I'm going to talk about in a second. These are like straightforward. So this is a microarray set. So we have donor nasal epithelial cell samples, which are the primary cell type infected by influenza A virus. And so we treat them with estradiol, or they were treated with estradiol and infected with influenza A virus. And then at 24 and 48 hours, they were assayed with microarray for gene expression. For the validation data, so this is work out of by Toki, where she starts with known of host proteins interacting with virus from LCMS. And using an siRNA screen and looking at virus titers and cell viability, we know which of those host factors are actually involved in virus replication. So the network analysis algorithm we're using is called DEMAND, so detecting mechanism of action by network dysregulation. And so there's a lot of simplifying assumptions here. So we start off with only genes that have like a one-to-one relationships between proteins that encode for, and we use like a protein-protein interaction database. And so we sort of prune off like genes that don't meet this criteria. And we also prune only to genes that are sort of expressed. So we have like the starting set of network interactions. And then we analyze the dysregulation. So sort of shown here in this C part of the plot is a scatter plot of these of replicates in the microarray. And we're trying to, and so we're measuring how much of this changed between our treatment and control conditions. And then we propagate these sort of significant changes, like back to a single gene and sort of control for those propagations. So what's kind of promising is using this method, we are able to uncover like more relevant, significant effects to our system. So we are able to both find better viral host protein interaction results, but as well as the viral replication host proteins. And of particular interest, we care a lot about this 48-hour time point of the effect of estradiol, because that's what the original microarray paper focused on most. And so my contribution onto the bioconductor side of things is more downstream of using this network analysis. So there's an enrichment, a trained enrichment database called TopGene, which runs a whole lot of enrichment analyses for you. And on top of that, because TopGene reports identifiers from different databases it's querying, I also use PubChem's ID exchange service to try and standardize those to PubChem IDs so you can analyze those further. And so this is just an example of some of the types of results that TopGene refers. So these are only 4 out of 19 categories. And so I know in Bioconductor, you do have bespoke packages that have put a lot of thought into trying to get you good results for these specific types of analyses. So this is just something you can do quickly. It takes 2 seconds for one of these queries, yeah. So that's sort of so this is sort of a technique that you can use to see, is there something interesting in the sample, especially if you have to deal with lots of samples. So in conclusion, hopefully what I've shown you is that network dysregulation analysis used as a primary way of trying to explore your data set of normalized gene expression actually has more value over trying to do differential gene expression and then doing network analysis after the fact. The demand algorithm I showed you, it's already in Bioconductor. It's been there for a while. Hopefully I'll have TopGene out for you guys also to use, but you can use it right now on GitHub. And kind of like a very stupid technical comment is, I think it would be really cool if we could kind of standardize using socket clusters when we parallelize instead of multi-core, because if your session memory keeps growing, you just use more memory, and that's kind of more consistent and frugal. And so yeah, I just want to thank especially our data, Sabra Klein Lab for getting us this wonderful microarray data set, and Toki for the validation. We have a new PhD student, Lauren Nichols, who had me annotate my DAM code and write lots of unit tests. So I thank her for that. And also I'd like to thank our funders at NIAID and the James Johnson Travel Fellowship for allowing me to present this work to you. No, yes.
So we have a couple of minutes available. Questions for Alexey? Yes. I'm going to bring you the microphone so remote folks can hear you.
Thanks for an interesting talk. You mentioned you've developed this for microarray data and this data set that you're working with. Are there scale issues that would come into play if you used RNA-seq information, or things that have vastly larger numbers of transcript and level information coming in?
Right. Yeah. So the network analysis doesn't take terribly long. So I think this was using 13,000 genes that kind of passed this criteria of being differentially regulated and represented in the protein database. So usually the limitation of using this method is the more replicates you have, the better. So there's a minimum of 3. I'm using 5 here, but so yeah, I think replicates are more important than anything else. I mean, I actually don't know what the computational complexity is for a number of genes, but I think the starting number of genes with a 1-to-where protein interaction with 26,000. So I think that's your upper limit, right?
Okay.
I don't know if I answered your question.
Yeah, I'm not sure if I understood it completely, but I'll chat with you later. Unless there's another question, maybe I'll just follow up with a different question about you mentioning you're working with genes, and I'm curious whether there's any value in working with transcripts or isoforms instead. Are you seeing any differential isoform pieces that might be masked by using the gene level?
Yeah, absolutely. So there are other algorithms. So yeah, I guess that. So then I guess then the question is, how are you trying to relate those isoforms to different interactions? So the difficulty is, if you have those different isoforms, you need to understand the relationship between, I guess, the proteins they're encode, and so you have like a more complex network. You could, I mean, if you had high confidence in these isoforms and had a network that had those high quality interactions. So one of the things you have to worry about is filtering for network interactions that you don't have a lot of confidence in, right? So if you did have that and you're studying a system in which you had that information, you certainly could do that. I don't think you could use this existing algorithm and do that. You don't have to do anything special for that.
Thank you. Hello, my name is Grace Kenny. I am a PhD student at University of North Carolina, Chapel Hill in the Fansdale lab. And today I'm going to talk a little bit about the R package I'm developing called Simple R. So studies in the GWAS catalog contain over 100,000 SNPs. About 31% of these overlap transcription factor binding sites. I think this is really interesting because transcription factor binding sites take up about 8% of the human genome. And so there's a massive amount of genetic variation that we can attribute, that could be really interesting that we can attribute to transcription factor binding, so much so that we can't really interrogate it all individually, too costly, too time intensive. So there's really a need for tools that help us kind of prioritize and investigate these SNPs on a large scale. So the goal of this project was to predict transcription factor binding, as well as to predict the effect of genetic variation on transcription factor binding. So you might be thinking, we're going to be looking at transcription factor binding sites across the genome. We might use some sort of motif analysis with position-weighted matrices, if you're familiar. And I think this is a fantastic analysis to do, but we thought it just didn't really fit our use case very well because we're more interested in calculating true binding propensity or affinity rather than similarity to consensus sequences. So instead, we looked to a tool called SEMPLE, or SNP Effect Matrix Pipeline. It was developed at the University of Michigan in the Boyle lab. And what SEMPLE does is it uses ChIP-seq and DNA-seq data to adjust position-weighted matrices to create what they call SNP Effect Matrices, or SEMs. Now, these SEMs structurally are very similar to position-weighted matrices in that we still have a base by position scoring system, but these scores are more indicative of binding affinity and less indicative of consensus sequence similarity because they are kind of adjusted and based off of that ChIP-seq data. Now, SEMPLE is a very fun and interesting tool, and it creates these matrices, but that's kind of where the functionality of this tool ends. It's a command line tool. It's relatively computationally intensive, and it doesn't really provide any tools or methods for further analysis or any visualization. And so my package is called Simple R, or SNP Effect Matrix Pipeline in R. And what Simple R does is it takes these SNP effect matrices, stores these in S4 classes. It stores multiple or collections of these SNP effect matrices in a collection class. It contains 223 of these matrices in our default data set, as well as some associated metadata. And these 223 default matrices really represent the vast majority of transcription factors that are most commonly interrogated, we believe. And so we believe that kind of using this default data set, most people could probably accomplish the analysis they set out to do, making it so you don't even necessarily have to touch that like really intense command line tool at all. So I'm going to cover very briefly a few of the functions included in the package. The workhorse of this package is really the score binding function. It just takes in a G-ranges object, SNP effect matrix object or collection of them, and a BS genomes object. And a little bit about how it works is, let's say you're really interested in that capital or that bolded A position. We can score a frame that includes that position against one of our SNP effect matrices. And because these are logged to normalized scores, we can just sum them up to create a raw score. And then we normalize that score based off a baseline that's produced by Semple. This baseline is kind of indicative of a background ChIP-seq signal. And we would get, for this example, a score of -0.65. Now, these scores can be positive, they can be negative. Negative scores, we tend to say, are low binding affinity and unlikely for the transcription factor to bind there. Positive scores, we say, are likely to bind. So the transcription factor isn't likely to bind in that position. However, there are multiple frames in which that contain potentially our site of interest. So we'll score all of those frames to find our top scoring frame. So it turns out that our transcription factor can actually bind to that site of interest. It's just kind of in a different frame because we get a positive score from that. The output is an S4 class that I won't dig super deep into, but probably the most exciting part of it is this kind of like scoring table that you get out where we do this process for all G ranges positions that you supply and every SNP effect matrix in the set that you use to create a score for like each combination. Now, there's a lot you can do with these scores, but something we really wanted to include in the package is some sort of like enrichment. So essentially asking, are some transcription factors bound more often than we would expect by chance? This enrichment function takes in similar inputs to the previous function, as well as a background or a null set. This background or null set you can supply by yourself, or we'll do some sequence scrambling on the back end to create one for you. And then we also include a plotting function to visualize the data. This plotting function looks something like this. On the exterior, I know it's really small and hard to see, but on the exterior are transcription factor names. The heat map on the inside is a -log 10 p-value of our enrichment test, which is just really a binomial test. And then on the very inside is a cladogram that is organizing the transcription factors based on the similarity of their SNP effect matrix. So you can see kind of like as we would expect, if we have like a transcription factor that's really enriched, we might see its neighbors show up as enriched as well. So that's why we're kind of seeing like clusters there, as well as kind of like a few others that are enriched in this data set. So I talked a little bit about predicting transcription factor binding, but I also want to touch on the effect of genetic variation on transcription factor binding. So essentially asking, is a motif broken, formed, or static with transcription factor binding? We have a similar function called score variance. that accomplishes this. And this really just gives us 2 scores instead of 1 because we have 2 alleles that we're scoring. We can visualize this because it's two-dimensional in like a scatter plot. And this scatter plot plots our 2 alleles binding propensity. So you can see our motif for that variant. Like I said, so the top left of that plot is going to be motifs that are potentially formed. Bottom right is going to be motifs that are potentially broken. And then anything that falls in between would be static. So like I said, we include 223 SEMs by default. So you can visualize all of them together. Similarly, you can visualize the effect of all variants on a single SEM. So this is just a conclusion slide detailing the 3 kind of things that this package can do. And then I'll wrap up by saying thank you so much to my lab, the Fengchao lab, as well as the collaborators at University of Michigan, my funding sources, and thank you so much to the JXTX Foundation and GBCC for helping me come to this conference.
Good afternoon, I'm Xintong from Genomic Research Center at FV. Today, I will like to share my recent work on developing a new R package for whole genome variant annotation, which is designed to streamline the association analysis of rare variant in large biobank. Genome-wide association study has been used in academia for almost 20 years. The traditional array-based GWAS have successfully identified many common variants, but their effects tend to be small. Rare variants, on the other hand, often have a larger effect, but they are harder to find and study. Whole genome sequencing enables a more comprehensive understanding of the genomic landscape by capturing both common and rare variants, including those in non-coding region. Genetics play an important role in pharmaceutical research. Genetics evidence supporting a drug target has been shown to significantly increase the likelihood of success in drug development. Here are 2 papers published in Nature Genetics and Nature News about the genetics evidence supporting drug discovery and development. The genetics research have now transitioned into the areas of large-scale whole genome sequencing. Here is a list of large cohort or biobank which whole genome sequencing data that I have worked on and currently are working on. Projects such like the UK Biobank have generated massive data set comprising about 1 billion variants, hundreds of trillions of genotypes, and petabytes of data. Text-based format such like VCF is the one of the most common format, not optimized for the scale of complexity of this data set. The genomic data structure GDS format addresses many of these challenges. GDS is specifically designed for the efficient storage and retrieval of large-scale genomic variant data. Compared with VCF, the data components are split and compressed separately in a GDS file. It integrates with R-based tools widely used in genetics epidemiology. For example, single relay genesis R package. Here, the UK Biobank whole genome sequencing data stored in GDS file is about 3 times smaller than that the peeling to pigeon file. Here, I'd like to introduce a GDS annotator, an R package designed for annotating variants stored in GDS file. It is optimized for speed and memory usage, making it suitable for analyzing millions of variants across the genome. It's integrated with the Bioconductor ecosystem. It also provides function for example, reformatting data from external database, including favors, nomads, and support saving annotation results from the popular tools such like Web and Snippet. and this package will be available in Bioconductor. A key integrate of GDS annotator is with favors. The favor database provides comprehensive pre-computed annotation for all possible SNP, as well as nodes insertion and deletion. The step pipeline is an analysis pipeline developed by a team of favors. To perform annotation informs real-world analysis. Favors annotation stop in GDS is about 35 times smaller than equivalent CSV5. Another example is known as GDS is 8 times smaller than VCF. In addition, user can convert the annotation from web and SNP to GDS for faster data queries and manipulation. Okay. Yeah. Here's a workflow I have been using for whole genome real-world analysis. It is a combination of the Sage GDS and GDS annotator package. Sage GDS is an R package I developed for pheno-wide association study using the linear and logistic mix model in large biobank. Table 1 lists the functional unit identified from the ensemble genome database. The protein coding gene are the most commonly used wide variant in, for example, long non-coding RNA, microRNA, and other units are also important. So the workflow here. So in the figure 1, at first, the genotype are converted to the GDS format, then conduct the whole genome annotation and convert throughout to GDS. Finally, run the Sage GDS package to perform set-based aggregate tests for the relevant. In summary, we are in the areas of whole genome sequencing. GDS is a store efficient for whole genome sequence variant data. A new R package GDS annotator is developed, and GDS-based pipeline enable large-scale whole genome sequencing association analysis. I also have a poster today. There are small details in the poster. Finally, I would like to thank my colleagues and collaborators at FV and Harvard University. Thank you for your attention. Oh, last one.
Well, wonderful. We have a couple of minutes for questions. I'm trying to remember now. GDS FMT is a CRAN package or a bioconductor package?
Bioconductor package.
It is bioconductor. Okay. Was there a question? I'm sorry. Oh, there we are. Okay, let me get you a mic. There's one back here. Okay, so you're.
Thank you. Great job. I have some questions. I work on rare diseases, and especially I'm now working, as you said, with genome files. So the problem now is, like, as you said, managing this data, this big data. And well, my first question is, can it be used in rare disease for rare disease annotation? I mean, I think, is it included like other databases like OMIM or whatever? And the second one is, what could it be? What's the difference? Do you think this program offers like certain advantages compared to other programs like Anovar and so on?
Okay, so your first question is to whether we can use the annotation for rare disease, right? So I think that's very common to use annotation for rare disease. That's just to what we did before. So the second question is, I think you see here I have pipeline. So if you use the pipeline and the R package there, then you can do the association by yourself.
Yeah, I have two titles. I couldn't figure out what to call this, so we'll see how it goes. I'm Aiden. I'm in the last week or two of my PhD at University of Pittsburgh in bioinformatics. I work mostly in comparative genomics. I build comparative genomics tools, and I wanted to talk about something I've been working on for the past little bit of time. So I want to start off with something that's probably a little bit obvious, which is that comparative genomics depends on us being able to compare genetic data. That's in the name, right? It's not to be cheeky or anything. It's just I think there's a problem that's a little bit underappreciated in comparative genomics, which is that we have to know what genes are even comparable in order to do any kind of comparative genomics analyses. Like knowing which genes are equivalent across organisms or not changes what we can do with them. So in order to do that, most of the pipelines that exist today do a variety of methods, typically involving a sequence similarity network where we have these nodes representing genes and these edges representing sequence similarity. Sequence similarity is like BLAST. It can be like MM-seq or Diamond, just some measure of how similar the genes are. Maybe there's pre-processing, post-processing involved, but we get this network. And from that, we use what's called a community detection algorithm to find groups of related genes. What I mean by community detection is we have some kind of network, and we identify groups of nodes that are more related to each other than they are to other nodes. Common things that you may have heard of to do this are like MCL, Louvain clustering, Laine clustering. These are common things. And pipelines, you typically see them in orthology detection pipelines. Okay. What is the purpose of my talk? Why am I building a tool? When we look at these sequence similarity networks, we start to notice a trend, which is that every organism has a genome, of course. Every genome has thousands, if not tens of thousands of genes. And as we build these networks larger and larger, every additional gene adds at least an edge per existing genome if we're using reciprocal best blast hits. Which is to say, in summary, this network will get bigger quadratically with the number of genomes. And so if you're using an algorithm that reads the network, the entire sequence similarity network into RAM, your RAM requirements will scale quadratically because you have to keep all of it in memory. Now, we talked earlier about how the cost of sequencing has gone down exponentially. That's really cool. Something I wish is also going down exponentially would be the price of RAM, but it is not. In fact, in the past 15 years, it's been basically constant. And a lot of people don't have access to these giant compute systems, or they're just really expensive. For the process we were trying to do, we just couldn't buy enough RAM to analyze the data we had. Something else we noticed, though, is that hard drives are pretty cheap and solid-state drives are continuing to drop in price. And so we were thinking maybe we could use some sort of like old-school computer science techniques to offload some of the data onto disk rather than putting everything into your RAM all the time. So this led to a new community detection algorithm for networks that I developed called XLabel. It's in the SynXtend package, which obviously is in Bioconductor because we're here. It's called that because it has two components. It has a community detection algorithm, which is called fast label propagation. It's like an extension of it. It's called XO because we put the data on disk rather than in RAM whenever possible. So it's sort of like building a database on the fly that we can query to get information about the nodes and use that to cluster this network. This means that instead of loading our entire sequence similarity network into RAM, we only need to load the number of nodes and some other data, and our memory scaling ends up being linear with the number of nodes and constant with the number of edges. In practice, what that looks like for this is on a large data set of synthetic networks. I'm going to sort of fly through results because I have a lot of them. So highlights for you guys. In terms of memory, you can see our algorithm in purple here and a bunch of alternatives all over the rest of the screen. And basically, we're scaling better. We have a lot less requirements. We can process a network that has around 31 million nodes with under 5 gigs of RAM and a single thread, which is cool. It's a nice fact, but obviously that's the case, right? I just told you we're not using RAM. We're putting it all on disk. Of course, we're going to have less RAM requirements than alternative approaches. Typically, the way algorithm optimization works is you're looking at trading off between three main components. There's not just low computational resources, but also accuracy and speed. Right, so if you have an algorithm that is, and typically we need to pick two, right? So if you have an algorithm that's accurate and slow, you could add multiprocessing maybe, you could add more RAM and make it go faster. If you have an algorithm that's really fast and accurate, you could use less computer and go slower. And so that's where we are. So I just told you I'm using low computational resources. Accuracy is sort of a non-negotiable when it comes to bioinformatics. So that was the next thing we looked at is not much RAM, but how well does it actually do? Our results are useless if they are bad. So we started with a data set that is semi-synthetic. There's other results as well, just these are the highlights. We used the known species tree. We simulated alignments on it. We built a network out of that. Details are here, but essentially, again, it's a network. Your nodes are genes. Your edges are either BLAST bit scores or BLAST PIDs. And the results are shown here. This is the largest screen I've ever presented to. We're this purple bar, and you can see, regardless of if we use bit score for edges or PID, we're basically one of the best algorithms, and we're consistent across either parameterization, so robust to different edge types, which is pretty nifty. If you take one thing away from my talk today, it should be this slide, which is that synthetic data is not real. So even if we're good on synthetic data, it doesn't mean we're good in the real world. So the next step was, can we do this on real data? And can we also have those sorts of same results? So we took every reference genome, prokaryotic reference genome from NCBI. We built the same kind of network. We tried to analyze it with all the algorithms. All of them crashed except ours. So we made subsets of it so that they could have a chance. And this is what our results look like. Again, there's a lot going on, but basically the takeaway you should have here is that relative to the industry standard of MCL, which is this red one, we have basically strictly better clusters. They are larger, and the genes inside them are more similar to each other than the equivalent ones in MCL, which is cool. Okay, so on semi-synthetic data and on real data, we're performing basically as good as alternatives, if not slightly better in some cases, which is nice. So sort of to wrap up, what's the damage on speed, right? I've picked the two things we're optimizing for. How much slower is it than competitors? We're looking at runtime on that last benchmark of those real data sets. Here's what it looks like. You can see up to the top, this is average runtime in seconds. And it actually turns out that we're about 3 times faster than the next fastest algorithm because of a lot of time spent optimizing. And if we extend that to all the graphs I showed in the first graph, this actually stays consistent across all of them. So we outscale them all. This is about a 3 times improvement. It's a log-log plot, so it's very small, but it's a lot faster. Again, this takes about 46 minutes, which is pretty fast. So sorry, I lied a little bit. In this case, in certain scenarios, you can actually pick 3. And that is what I'm presenting here. It's an algorithm called XLabel. It has less RAM requirements, less processor requirements, about as accurate as competitors, and it's faster. So if you need to do network immunity detection, it does that. There's not a lot of time for questions. I wanted to sort of head off the ones I get every time I present these to save some time for you guys. Can you use this on your favorite graph of choice? Yes, you can. It does not have to be a sequence similarity network. This is what we care about. You can use it on other stuff. I sort of ignored all the storage we're using, like the disk requirements. It's usually the same order of magnitude, around 1 to 2 times the input file sizes. You can ask me about that if you're curious. People always ask me about the back end, if it's DuckDB or if it's Arrow, or if it's another thing that they really like. I can tell you it's none of those for sure because it is just built from scratch in R. It's got also C. And then I would love to talk about the computational details, but I only have 10 minutes. So you can find me and I will talk your ear off. Or you can look at this QR code if you don't want to talk to me. This is my lab. This is my website. This is our lab's website. This is our package. My advisor maintains Decipher and I help maintain biostrings. Feel free to talk to me about whatever. Thanks to my funding sources. Almost forgot.
That's fine.
Thanks.
One question?
Yeah. Oh, I guess we need a mic. Sorry.
Yeah, so I know you've put a lot of effort into the optimization, but is there like one big thing that you'd say is that you found very useful or you'd recommend? Is it thinking about CPU caches and struct optimization?
Yeah, no, it's a terrific question. Honestly, it's just disk access patterns. That tends to always is the bottleneck, and the amount of time you can save by like sequential read and write and reading in blocks is immense. It's like a 20x improvement when I switched from reading in sequence versus reading and writing in random.
And so can you give us an idea of how much this does it use? Like, does it does it not necessarily need to use a shared file system? Like, can you do it all local or?
Good question on how much, can you clarify? Sorry.
Well, so shared file systems are really garbage in terms of consistent performance, right? It just
That's true.
depends on other people, what they're running. And so just kind of.
Yeah. I haven't tested this with
I don't know how much disk space you need basically. Also.
Right. I haven't tested this on network storage. The disk space requirements tend to be around 1 to 2 times the input file sizes. So it's not insane. You should be able to do it locally if you can have the graph in the first place. But going towards that would be interesting. We're looking at ways to sort of scale it from big to big.
Thank you.
Right.
Sorry.
My name is Lambda. I'm a postdoc at Columbia, and and I work with Bioconductor, and today I'm going to talk about how I used Voyager to perform a multi-scale exploratory spatial data analysis, or ESDA, on a lung carcinoma dataset. So I'll talk about univariate, which is spatial autocorrelation, Morris I here, and bivariate, which is for co-expression. So like that's least out, and the multivariate ESDA here on this data set. And I'll touch a little on the I'll touch a little on the how these results would can vary across stages of LUAD. So as we know in biology, there's a very wide range of spatial scales, from ecosystem all the way down to tissue, cells, and molecules. And at each of the scales, there can be spatial regions. Also, for the technologies used to quantify gene expression in space, we also have a wide range of spatial scales. So here we see so that's only one Visium spot, and so Xenium is single cell resolution, which is shown here, and so we can see how these are how the many of the other technologies compare to Visium in terms of the spatial scales. And in addition, we have for those technologies we have different detection efficiencies, and there's also sequencing based technologies which are transcriptome wide as opposed to gene panel for usually a few hundred genes for imaging based but single cell resolution. So there are many. So people often apply the same analysis methods to data from these different technologies with different scale and different besides spatial resolution, but also other technical differences. But I've been thinking wouldn't the results mean different things, and what can one technology find that the others cannot due to their differences, and especially what are the spatial things that we can find with say Xenium or Visium that the other cannot find. So to answer this question, I searched for studies with data from both Visium and Xenium from adjacent sections, so I can align them. And so I found one from for lung carcinoma or LUAD, and because they're from adjacent sections, I just used the histology to align them. And in order to find detection efficiency as opposed to spatial resolution, I made a pseudo Visium. So basically, I used the Visium spot polygons to aggregate the Xenium transcript spots. So basically, just to count the number of transcript of each gene that falls into each of the Visium spots. And I performed ESDA on those. So this is a very comprehensive study. It has so I have samples from multiple patients across different stages of LUAD. So the earlier stage is AIS, and the later stage will be invasive. Morris I is the most commonly used method in ESDA. So it's a metric for spatial autocorrelation, and so here in this plot, I plot Morris I for each gene in Visium in X, and the Xenium or pseudo-Visium in Y. So generally in Xenium and pseudo-Visium, the Marrah is higher because of the higher detection efficiency. So an example here will be a gene bank one, which is pretty much like there's no spatial structure in Visium. It's very sparse because of the dropouts, I suppose. So like it just completely killed the pattern that we see in Xenium. And so the square points are for pseudo-Visium and the round one for Xenium. And so for so in many cases, so spatial aggregation increased Marrah's I. Yeah, so that's when with the square ones on top. But there are also many cases where it's spatial aggregation decreased Marrah's I. So that's with the round ones on top. And so to see some example, this is an example, ApoE. So in this case, after spatial aggregation, Marrah's I decreased because this gene has a lot of spatial structure, but the length scale of the spatial structure is much smaller than the Visium spot. So it vanished after it's vanished after aggregation. And there's another gene here which drastically increased. So basically, at a single cell level, there's a lot of heterogeneity, but after aggregation, like we get to see the larger scale structure better. So I did the same for least L. So it's like a spatially informed kind of correlation. So to read this plot, so like a blue color means the cell expresses is high in this gene for marker for metastasis, but low in the other one, and the yellow's high in this one, but not in this one. And so the green means that it's high in both. So again, Visium, the sparsity killed pattern, but in Xenium, so we see there are many blue and yellow cells. There's a lot of heterogeneity, but after aggregation, at least the correlation increased because there are separate cells that expresses either of those, and they came together into the same neighborhood. So at this pseudo-Visium, we were actually analyzing the neighborhoods instead of the cells. But why stop at Visium versus Xenium? So I started this project on Visium versus Xenium, but I can go further. So what about investigate like what happens with the spatial scale for like a wider range of scales? So I used the SFR package to generate those spatial grids of different sizes, and I used the spatial feature experiment package to aggregate them. And so the output is stored on disk with Alabaster. And so like again, I computed those ESDA things for each of these resolutions. And for Marrah's I, so turns out there are different patterns of how it changes with scale. So there are many that increased, just like in one of the examples, and but there also like just as in the examples, there are some that will peak, and so some would decrease but with a peak, but there are some that are flat. And I have also recapitulated these with some synthetic data. So here are some examples with real and synthetic data. So it increased because of the sparsity and the local heterogeneity, but later on it decreased because the pattern gets smoothed over. So I did the same for least L, like that's a gene correlation. So again, I see different patterns. There are some that increase, there are some that peak, and but interestingly, there are some that will become more negative after spatial aggregation. So why? So against this by scale plot, and so here, so again, when it increases, it's because like there are neighboring cells that come together as a neighborhood. So like we're really analyzing at the neighborhood level instead of cellular level, and so we get different patterns. But so here when it becomes negative, it's really that there is a larger scale spatial structure that's not obvious at a lower at higher resolution. And for least L, we actually need both genes to be spatially structured in order to deviate from 0. And also did like multivariate. So basically, that's how like this is like a spatially informed kind of PCA, and how the loadings. This shows how the loadings change, and again, basically at a small scale, so we can distinguish from between alveolar cells and immune cells, and that explains most variants here. But after aggregation, they came together into the same neighborhood, so we can no longer so this can no longer explain the variance. So how will this change? So basically, in here, like in some cases, actually will change with stages of cancer. Like here, this example indicates the metastasis, and that's probably with the neuroendocrine cells. And yeah, so there's a package called Wayfarer and still on GitHub, and I'll work on submitting to Bioconductor, and yeah, those are the acknowledgements. Thanks.
Okay, super. Sorry for the rush. One question since we had some catch up.
Go to the poster session so we can talk out the posters.
We do that. Thank you so much.
Yeah, welcome.
So my name is Astrid Deschênes, and today I'm going to showcase the bioinformatic workflow we use to decipher CLEC12A negative regulation of neutrophil activation. First and foremost, this is a collaborative work, and the leader is Maria Fernandez, as a laboratory is studying CLEC12A when there's arthritis in patients with arthritis. So what is CLEC12A? It's a protein in this figure. It is the protein in the center in pink. It is located on the membrane of the cells, and it is an inhibitor receptor that negatively regulates neutrophil activation. In the case of rheumatoid patient, when there's a low CLEC12A cell surface expression, there is a higher disease activity. So what we try to answer in this project is what are the molecular mechanisms that are employed by CLEC12A to negatively regulate neutrophil-driven inflammation. So the workflow is not that complicated. We start by creating a protein-protein network with specific filters. Out of it, we create a CLEC12A-centric network, one containing only the first-degree interactors and one containing the second-degree interactors. From this latest network, we add a phosphoproteomic dataset, and we annotated to create a subnetwork. And for all those three networks, we run functional enrichment, network analysis, visualization. We extract potential novel insights that we validate with the target experiment. So the global protein interaction network, it's been created using a BioGrid, which is a public database. We apply specific filters, such as human data only. We remove chaperone proteins which interact with a lot of proteins, and we use specific experimental approaches. We also ask that for the second-degree interactors, we have at least 2 citations. So out of this global network, we build our first-degree CLEC12A-centric network and our first-degree and second-degree interactor network. So this is our first-degree network. It's relatively small, and what is interesting is out of those 32 primary interactors with annotation, we notice that 12 are involved in vesicular trafficking, and 4 are signaling proteins. So the vesicular trafficking is in dark blue, and they interact a lot with each other, and the signaling are in red. So we try functional enrichment. We did not know if it would work because it's a small data set. And fortunately, we had 88 significant results using gProfiler package. Those are all the results. And among the top results, we have the snare complex, which is associated with the vesicular trafficking. So we use our package enrich unit to generate a functional enrichment graph where the nodes are the term of interest and they are linked when they share at least 20% of their data sets. And out of it, we found two modules of interest, the signaling module and the vesicular trafficking module. So all those results are consistent and highly interesting. When we move to the second degree network, this network is way larger with more than 4,000 secondary interactors. And the functional enrichment gave more than 2,000 significant results out of it. I'm just showing that we still find the vesicular trafficking module, which is a bit smaller. But in addition, we find this large, I did not show it all, but it is a kinase activity module. So we take this network and we decided to annotate it with the phosphoproteomic dataset that we had available. This dataset, it points to the protein whose phosphorylation is under the control of CLEC12A during homeostasis in inflammatory stage. And when we annotate and we retain only those protein, we end up with a smaller network with 39 secondary interactors. And what is interesting from this network is like 10% of the secondary interactor are MAP kinase, and MAP pathways are associated with inflammatory response. So all our results are related and seems to be biologically relevant. So out of all those results, we decided to run a targeted experiment. So I'm just showing one of the experiments where in Marrah's lab, they generated a CLEC12A overexpressing neutrophiles, and they look for specific MAP kinase. And in this situation, in the overexpressing CLEC12A neutrophils, you see that phospho-ERK1 and 2 is completely almost absent. It's really downregulated. So this is the ratio of phospho-ERK overexpressed versus wild type through time. So that was a very interesting finding, as ERK pathway is a subpathway of the MAP kinase pathway family. In conclusion, mining public data set can lead to new insights. It's not really that expensive. It's even better when you have your own data set that you can incorporate it. And it's also very important to confirm your findings through either biological experiments or orthogonal experience. In the case of CLEC12A, we find that vesicular trafficking might be a potential mechanism that explain the CLEC12A negatively, or CLEC12A negatively regulates neutrophil activation. And we are certainly going to study a bit more the ERK protein and the ERK pathway. I have an associated poster that's going to be shown tonight, if you are interested. The future direction is mainly, we have started writing the paper. Hopefully it's going to be out this year. And we are still working on improving the bioconductor enrichment package. And I would like to finish by acknowledging the Canadian government for giving us grants for this project. Thank you.
Good. It's still in green, so if anyone has a question, we have time for that.
Yeah.
Yes, break again. Okay.
Hi. It's a very cool talk. So you mentioned that for the first degree interaction network, you're looking for at least 2 citations. And I know it's like a heroic effort to like develop these sort of hand-tuned networks, but for the secondary network for those, like, do you have any recommendations for how to like create like a, high confidence network for those interactions? Like,
So we work a lot with the biologists on our teams, and they are the one who pick up the experiment that we should retain, that they were the most confident. And we look up at, for the minimum citation, it was also looking at how it would shrink, and at each time, like the biologists would look at the result and see if something makes sense or not. Like the chaperones, they were like, at first we did not remove them, but it was of no interest to have chaperone-related results in our analysis. So,
all right.
Very nice.
I'm assuming this is high enough. There it goes. All right. Hi, everyone. Thanks for having me. My name's Tyler Collins. I'm going to be telling you kind of about a larger project that's been working in association with Galaxy called BioDigs, as well as a lot of the new tools and workflows that have been introduced into Galaxy as a result from the project and some of the ongoing work that's currently happening with it. So BioDigs is kind of based upon two pillars for what the project's trying to achieve. The first is a better understanding of microbial diversity within soil. Oftentimes, soil is a pretty uncharacterized environment. It's often predicted to have a significant amount of biodiversity within it. Multiple papers have made the argument that it's potentially the most biodiverse habitat on Earth, yet very often most of the species in it can't be cultured either in a lab, and there's surprisingly little sequencing on it compared to many other organisms and environments. In addition to this focus, BioDigs is also partnered with the GDSCN, which is the Genomic Data Science Community Network. It works in order to expand science and outreach to lower resource institutions across the United States. So very often these are your historically Black universities, tribal colleges, community colleges, and engage with the students there and have them engage with institutions with higher levels of resources and to be able to perform meaningful biological analyses and hopefully get published. So these two efforts combined sort of led to the formation of the BioDigs project, which is both focusing on developing methodologies and ways to research that microbial dark matter in the soil and ensure that it can be used and participated with these different institutions. So to that end, a sampling protocol was developed that engages them. So right now we essentially create individual kits that have different chemicals and DNA fixing agents that they'll need. We're able to send them off to all these different institutions across the United States. They go out and sample soil. That is done in multiple replicates in multiple different sites. Once that data is collected, or the soil is collected, it is shipped back across the United States. It is sequenced or tested in Delaware for lead, heavy metal concentrations, pH, other metadata. And additionally, it goes through a variety of sequencing. That sequencing is carried out at Cold Spring Harbor, as well as Johns Hopkins itself and the LabCorp. We sequence every single sample with Illumina sequencing. In addition, a fair number of the samples are also sequenced with PacBio as well as Oxford Nanopore to try and create a pretty comprehensive amount of long-read sequencing data for future testing and for further analysis. This data is then uploaded into Galaxy and shared across everywhere, and we kind of continue with our analysis. And so to kind of begin with what we're looking for to pull out of this, one of the very first things, one of the first analysis you do is some form of taxonomic classification. This can be done on the individual raw reads that you pull from your sequencing instrumentation. What you're seeing here is just a plot generated by Kraken2, which is a very common sequencing methodology here that was run on one of the samples from the Montgomery County area outside Washington, D.C. And in this first one, you're not seeing that much of interest. Most notably, you'll see several species that are commonly associated with nitrogen fixation, pretty normal species. You'll have a couple others that point out that are often associated with like naturally occurring antimicrobial elements, or that have some association or importance potentially with humans. But it was pretty early on that we realized that this was an approach that wasn't particularly effective. When you actually are looking at what Kraken-2 is classifying with its k-meray approach, a majority of the reads are returning as blank and empty hits with no actual meaningful information. So from that entire assembly we built, only about 12% of that sample was actually able to be classified across all the k-mers, and the vast majority of it was unknown. This is against the largest database you can use for Kraken right now, which is the, if I remember right, it's the GBTK database. It's about 600 gigabytes you have to load into RAM in addition to all of your samples to run, to actually run through the whole thing. It's, to my knowledge, the most currently comprehensive list of all known species that could be in the soil. And even with that, we're still barely getting any classification. So one of the first things we looked at and tried to improve upon is a different method that we could add into Galaxy to both make available to people, as well as hopefully get better results. And so we started working with SILF. SILF is another kind of profiling and containment estimate software with a slightly different approach. It essentially does a subsampling of k-mer reads and does an ANI-based approach. Essentially, the big win for us is it significantly reduces the amount of RAM you have to use for reading things into memory, even across the same database. Additionally, it runs significantly faster and it allows for slightly less sensitivity. So you're actually able, if something's not an exact match with a k-mer, which is often the case in highly diverse environments like soil, where it's very complicated, especially with our sequencing depth, you're still able to get pretty meaningful results. And so running through SILF, this is a subsample of about 50 sites we've done through Baltimore and Montgomery County out of the over 100 we have done in total. You're actually able to classify about anywhere from about like that 15% you were getting from Kraken-2 up to about 50% are being able to successfully classified. We've been doing further validations, additional steps on other classification methods. But at this point, we have SILF fully available in Galaxy, and it's fully able to be, I don't think it's on main actually right now. I think it's in the Galaxy toolshed, but it's able to be run and can be replicated on the samples. So in addition to things that need to be added to Galaxy to try and enable these analyses was sort of an approach on MAGs. So very often in these kind of analyses, the contig level resolution is not enough. So you have to do some kind of like metagenome assembled genome with some kind of binning step. This is in reference to a paper from a database we had been using that was essentially looking at about 40,000 soil specific MAGs. We compared our own data against this and found we wanted to try and do a different methodology to assemble our own. So we sort of focused on the PacBio Revio reads we were working with and have implemented this MAG analysis pipeline. So essentially, you are doing some kind of assembly of your contigs, usually in MetaMDBG, which is a long-read aware assembler that works well on metagenomic reads. At that point, it gets split into mostly complete contigs that essentially get saved as long as they are assessed to be above 90% completion, or if they're below that 90% completion threshold or shorter than 500 KB, it goes through a series of binning algorithms. So it runs through MetaBat2, SemiBin2, and then kind of a combination step that evaluates them with DOS tool. These are usually pretty effective. SemiBin2 can take a while. It uses a different like machine learning models, and you can train it on your own data. Very often we've been using just the pre-trained soil data. But once you get all these, you dereplicate your bins, you do some kind of final assembly check, and at the very end, you get out a nice fast day with your MAGs that can be used elsewhere. On the right is sort of just one of the runs we had done on one of the small samples where we were trying to compare this MAG pipeline compared to the short read one we were using, where from using this, we were able to get significantly more MAGs coming out. And so as I can see, I'm running out of time. Currently, we've taken that MAG pipeline that we had in SnakeMake, and it is not fully implemented into Galaxy yet, but this is kind of one of the first drafts of it going into Galaxy. Of trying to ensure that will be available, it can be shared. I'll briefly mention In addition to all this ongoing work, we also have several other working groups working on this project, specifically looking at metal efflux pumps and environment associations, as well as health and human disease relations. And then all this data is also now available in the Galaxy Data Library. And with that, I just wanted to thank everyone in the community.
Hi, so I'm going to talk about Explain, which is a Shiny app that we have developed to visualize the results of differential transcript usage analysis. So first, I'm just going to explain the type of data that the app takes, and then just showcase the app. So to put everyone into context, as we all know, when we have in eukaryotes, when we have our pre-mRNA molecule, there has to happen splicing in order to obtain our final mRNA isoform. And we also know that not all the exons are always constitutively spliced in. So it happens alternative splicing and this can happen in many different ways, but I'm just going to explain a little example of how this can happen. So we could have an RNA binding protein that binds in an intron upstream region of an exon that is going to be regulated, that is going to be downregulated in this case, so that in our final isoform, we don't have this exon present. If we were to do an experiment where we knock down this RNA binding protein, we would have a final mRNA isoform with the exon present in this case. So if we were to analyze the proportion of the isoforms that we have in these two experiments, we can see that in the wild type, or when we have our RBP present, the predominant isoform is this first one. And then in the knockdown condition, the second isoform is the one that is more predominant. So we can see that there is a DTU event happening in this gene, but like what we are interested in is like why happens this. We are wanting to find a connection of these isoform changes with their downstream functional consequences. So we can think about questions about what are the most common isoform structural changes that are happening out of all our data set, which genes show DTU or differential transcript usage, if the isoforms that we are detecting, are they protein coding or are they non-coding? And if they are, which domains of the protein are being affected by these isoform changes. We can also think about how did that even happen in the first place. So about how the RBP works in the first place. So what is their binding profile? What type of sequence is it recognizing? And also in which context this RBP is getting expressed or repressed. So to put a name, something more specific, we're going to work with PTBP1, which is an RBP that works exactly as I just showed. So it's a polypyrimidine track binding protein. So basically it downregulates exons when it recognizes sequences that are rich in U and C. And we are using this because we have a long-read RNA-seq data from the genotype tissue expression consortium that basically have these two experimental conditions that we just talked about, a wild type or with PTBP1 or a knockdown of this protein. So when we have long-read RNA-seq data, this is the typical workflow that we can see that we would go to do and explain basically after we have done assembly, identification, quantification at the DTU, we are going to want to use the app for detecting exon level events and just in general visualize our DTU results in a user interactive and friendly way, and in a way that it modularly fits into this bioconductor ecosystem so that it doesn't matter which tools you have used to get into the DTU, you will be able to run the app. So I'm just gonna explain very briefly how we would do the skipped exon event detection in spleen. So basically we need two isoforms, one that is more likely to be expressed in presence of PTBP1 and one that is less likely to be expressed, more likely to be expressed in absence of PTBP1. And we consider if we overlap these isoforms, candidates are those that are not present in the wild type isoforms. And we consider that these candidates are actually downregulated exons if we look to the upstream and downstream exons, and we find them in both type of isoforms. So explain this in the back, and now I'm just gonna show a little bit of how the app actually looks like. So in the main panel, oh, hello. In the main panel, we have a DTU table, which is basically your result table. Then you can click around and your transcript is associated with a gene. And in this table, it will just tell you which gene it is and a little bit of its ontology. And then in the main plot, we will visualize the isoforms that are differentially used for this gene. In red, it would be those that are more likely to be expressed in absence of PTBP1, and in blue in presence of PTBP1. And then just to see the quantity of the change, we can see the proportion and the quantification of actually how big that change happened. Apart from the main tab, we have also like an exon level tab where we have our same DTU table, but also we can visualize if we have detected an skipped exon event or a downregulated exon event, we will be able to visualize that if that has been found for a specific gene. And we can visualize this gene by gene, but we can also see, since explain does this for all the genes in the data set, we can visualize the upstream intronic region of these downregulated exons, and we can see that for the ones that we detected, there is a very strong signal of a higher percentage of C and U that matches what we just explained at the beginning, that this PTBP1, this RBP binds to, and is different from those exons that we are considering to be non-regulated by PTBP1. This is just a little gift to show that it's just a shiny app, it's interactive, you can play around, change the filters for your data, and just a little bit of exploratory after you've done your analysis. So to wrap up, I just want to briefly say that Explain is a very early work in progress. It's not a package yet, it's just here in the GitHub repo if you would like to try. We can so far interactively visualize at the isoform and the exon level some events, but we still need to include many other type of exon level events. We also would like to include a functional classification of these isoforms and analyzing how the protein domains are being affected if they are coding, and then just keep improving this modular integration into Bioconductor. And I just wanna, this is my acknowledgement, I just wanna thank especially my lab that I just recently joined. Thank you so much.
Super. Any questions? Again.
Very cool talk.
Thank you.
So like about 1% of the total cell RNA is being actively transcribed. So it's kind of curious, how do you, I mean, you could have total RNA-seq, but then if you're doing long read, you know, you only have so many pores if you're using Oxford Nanopore, you don't want to waste those. So it's kind of curious, how do you enrich for the actively transcribed, you know, co-transcriptional transcripts?
For the quantification?
Yeah.
So that is a great question that I don't work on that. So like basically where I take over, it's basically all this workflow, right? I think that would be like a question or like a good thing to think about when we're doing like the quantification step. I haven't really like studied that yet, but that would be great for whoever develops these kind of methods. Thank you. The one
How do you do the knockdown is the question.
experimentally. I believe it was a generic knockdown. We took just to develop the app, this public data set. I can look into how they actually did the knockdown experiment, but yeah, it's obviously.
I think that's a good point to wrap up on.
Maybe a good conversation for the wine. Yeah.
All right.
Okay. Thank
Once
you so
again,
much.
let's thank all the speakers. Thanks for ending on time.